Imagine a spark traveling down a wire, but far smaller, measured not in volts you can see, but in whispers of charge across living cells. How do these quiet pulses become the foundation of every thought you have? Welcome to Neuro Rest, where we explore the essentials of neuroscience in a calm, structured way. Today we'll trace the path from single neurons to vast networks, revealing how circuits coordinate perception, memory, and decision. To make this journey complete, I've prepared a free practical guide with a concise summary of today's lesson, waiting in the description like a map you can return to whenever needed. Settle in, let's begin where science meets the living mind. A single neuron is so small you could not see its working parts without magnification, yet each one carries the same principle as a spark running through a wire. In every brain, billions of these sparks are in motion at once, and it is this constant activity that makes thought, memory, and perception possible. To begin, imagine standing in a quiet field at night. A sudden flash of lightning appears in the distance. That flash is short, it carries energy, and it marks the beginning of something larger in the sky. Neurons operate with similar flashes, though their storms are not in the clouds but across a thin membrane that separates the inside of the cell from the outside. This boundary is alive with electrical differences, with the inside of the neuron kept more negative than the outside, a state known as the resting potential. It is like a loaded spring ready, but not yet released. The architecture of a neuron is designed to handle this energy. At the center is the soma, the body of the cell that holds its nucleus and sustains its life. From this soma stretch delicate branches called dendrites, shaped like trees in miniature, which receive signals from other neurons. Extending in the opposite direction is a long axon, a cable that carries signals away. The axon may be wrapped in insulating layers known as myelin, which speed conduction, allowing the signal to travel farther without fading. At its end, the axon divides into terminals, where information is passed on again. This division of labor, receiving, integrating, and sending, gives the neuron polarity, a clear sense of direction for its signals. The spark itself is known as an action potential. It begins when the neuron is sufficiently excited by its inputs, crossing a threshold. At that moment, ion channels embedded in the membrane open rapidly. Sodium ions rush inside, followed by potassium leaving in response. These movements change the voltage across the membrane, creating a wave that surges down the axon. The action potential is not a matter of degree. It either happens fully or not at all. This all-or-none property ensures fidelity, the same kind of pulse, repeated and reliable from one end to the other. Why does this matter? Because the brain's language is frequency, not amplitude. A neuron can fire more or less often depending on the strength of the stimulus, but the shape of the action potential remains nearly identical. A faint touch on your skin may produce only a few spikes, while a firm pressure will send many in quick succession. This frequency coding allows the brain to represent intensity without confusing it with type. Once an action potential reaches the end of an axon, the story changes. Here, electrical activity becomes chemistry. Tiny vesicles filled with neurotransmitter molecules wait just beneath the membrane. The arrival of the spike causes calcium channels to open. Calcium flows in, and the vesicles fuse with the surface, releasing their contents into the synaptic cleft, the narrow gap between one neuron and the next. Neurotransmitters drift across this space and bind to receptors on the dendrites of the neighboring cell. If the effect is excitatory, the receiving neuron becomes more likely to fire its own spike. If the effect is inhibitory, it becomes less likely. In this way, neurons whisper to each other, a constant conversation of encouragement and restraint. The balance between excitation and inhibition is delicate. Too much excitation, and circuits run into overdrive, as happens in seizure activity. Too much inhibition, and signals may be muffled to silence, dulling perception and slowing action. Healthy brain function requires a finely tuned mixture of both, adjusted moment by moment depending on context. In some regions, like the cortex, inhibition sharpens edges, helping you see contrast more clearly. In others, like the spinal cord, inhibition prevents opposing muscles from contracting at once. Everywhere, it keeps the system from chaos. Alongside neurons stand glial cells, once thought to be mere glue, now known to be active partners. Astrocytes help recycle neurotransmitters and maintain chemical balance. Oligodendrocytes and Schwann cells create myelin, speeding transmission. Microglia patrol for damage and infection, pruning unnecessary connections. Without these glial companions, neurons could not thrive. They are caretakers and regulators, ensuring that sparks can run smoothly without exhausting the system. Even at this microscopic scale, patterns begin to appear. Two neurons connected in sequence form the simplest circuit. Add a third, and you can create feedback, amplification, or inhibition. Multiply this across thousands, and you arrive at motifs, common arrangements of neurons that the brain reuses in many places. Feed-forward loops pass signals quickly. Recurrent loops sustain activity. Lateral inhibition enhances contrast. These motifs are the building blocks of larger networks, and from them arises the complexity of perception and thought. One important principle of circuits is plasticity. When two neurons fire together repeatedly, the connection between them can grow stronger, a rule often summarized as cells that fire together wire together. This is the essence of learning. But plasticity has boundaries. Synapses can strengthen only so far, and unused ones can weaken or disappear. Plasticity is not unlimited. It is shaped by development, by environment, and by experience. When you focus your attention on something, these principles come together. Neurons in the visual cortex amplify certain inputs while suppressing others, guided by signals from the frontal lobe. The result is like a spotlight in a dark room. One area becomes vivid while the rest fades. Attention depends on excitation, inhibition, and plasticity, working in concert, allowing the brain to select what matters in a sea of possibilities. As we close this opening section, hold the image of a single spark, carried down an axon, crossing a synapse, and igniting the next cell. From this simple event, so small it vanishes in less than a blink, arises the vast orchestra of the nervous system. In the next section, we will step deeper into the machinery of membranes and channels, the hidden gates that make this spark possible. Every neuron depends on a boundary that both separates and connects it to its surroundings. This boundary, the membrane, is more than a barrier. It is a living interface that decides which molecules may pass and when. Without it, the spark of an action potential could never arise. The membrane is thin, only a few molecules wide, yet it holds back charged particles in careful arrangement. The outside of a neuron is rich in sodium and chloride. The inside contains more potassium and organic anions. This difference is like water behind a dam. The potential for flow is always there, waiting for the gates to open. Those gates are ion channels. Some open in response to voltage changes, others respond to chemical messengers, and some remain open at rest to maintain balance. Each channel is a protein folded into a precise shape, creating a pore through which specific ions may travel. One may allow sodium but not potassium. Another may admit chloride but exclude calcium. This selectivity makes the channels gatekeepers of electrical communication. Consider voltage-gated sodium channels. They remain closed when the neuron rests. A small depolarization nudges them, and once threshold is reached, they snap open. Sodium rushes in, driven by both electrical attraction and concentration difference. The influx further depolarizes the membrane, causing more channels to open in a chain reaction. This positive feedback produces the rapid rising phase of the action potential. In milliseconds, the inside of the neuron becomes more positive than the outside, but the surge cannot continue unchecked. Almost as quickly as they open, sodium channels inactivate, refusing passage until reset. At the same time, voltage-gated potassium channels open, allowing potassium to exit. This outward flow brings the voltage back down, a process known as repolarization. Often it overshoots, making the inside even more negative than usual, a stage called hyperpolarization. Then gradual closure of the potassium channels and the steady work of pumps restore the resting potential. This cycle depends not only on the channels but also on a molecular machine, the sodium-potassium pump. Working constantly, it ejects sodium from the cell and pulls potassium back in, consuming energy with every cycle. Though slower than channels, its persistence ensures that the gradients are maintained. Without the pump, repeated firing would dissipate the differences, and neurons would lose their ability to signal. Calcium channels add another layer. Found at axon terminals, they open when an action potential arrives, allowing calcium to rush inward. Unlike sodium and potassium, calcium is scarce inside, so its entry is a powerful trigger. Vesicles sense this change and release neurotransmitters into the synaptic cleft. Thus, calcium links electrical events to chemical communication, bridging one neuron to the next. The precision of channel function is vital. If sodium channels fail to close properly, neurons may become overactive, leading to seizures. If potassium channels are faulty, repolarization may be delayed, slowing response. Mutations in calcium channels can impair neurotransmitter release. altering communication throughout circuits. Many neurological disorders can be traced to such channel dysfunction, sometimes referred to as channelopathies. Pharmacology has long taken advantage of these gates. Local anesthetics, for example, block sodium channels in sensory neurons, preventing pain signals from reaching the brain. Certain toxins from animals like pufferfish also target sodium channels, but with dangerous consequences. Potassium channel openers can reduce excitability, while blockers increase it. Understanding channels at the molecular level has opened doors not only to medicine, but also to deeper insight into how neurons perform their work. Beyond individual channels lies their coordinated action. In dendrites, ligand-gated channels respond to neurotransmitters, generating small changes in voltage called postsynaptic potentials. Some are excitatory, bringing the neuron closer to threshold, others inhibitory, pushing it further away. The neuron sums these influences across its surface. If the combined effect is sufficient, voltage-gated channels at the axon hillock launch an action potential. This integration of countless inputs into a single output is the core of neuronal decision-making. One can think of the neuron as a voting system. Thousands of synapses cast their ballots through channels, some urging fire, others urging remain silent. The outcome depends not only on numbers but also on timing and location. Inputs arriving close together in time may combine, while those arriving on distant branches may fade. The axon hillock, with its dense cluster of sodium channels, acts as the final tallying station. What makes this machinery extraordinary is its speed. Gates open and close in thousandths of a second, yet in that brief interval entire chains of events unfold. Neurons can fire repeatedly, carrying streams of information across the nervous system. Though each channel is small, together they form a dynamic landscape, always shifting, always responsive. The existence of such gates and gradients raises reflection. Why would evolution choose to rely on charged ions rather than some other signal? The answer may lie in efficiency. Ions are abundant, their movement is fast, and electrical differences can be set up and erased with little structural change. A neuron can fire endlessly without consuming its architecture, relying only on constant supply of energy to maintain the gradients. This makes the system robust yet adaptable, capable of rapid signaling over a lifetime. As this section closes, hold in mind the image of gates in a dam, opening to let torrents of water rush through, then closing to restore balance. Neurons do the same with sodium, potassium, and calcium, crafting momentary surges that allow thought and perception. In the next section, we will follow these surges into the phenomenon of the action potential itself, where the spark becomes a traveling wave of communication. The action potential is one of the most elegant events in biology, a fleeting surge of voltage that sweeps down the axon like a traveling spark. Though brief, it is the very heartbeat of neural communication, repeating endlessly across the nervous system. Without it, no signal could move farther than the reach of a dendrite. With it, the brain becomes a living network, capable of transmitting information across vast distances within fractions of a moment. At its core, the action potential is a self-propagating wave. It begins locally, often at the axon hillock where the density of voltage-gated sodium channels is greatest. When excitatory inputs depolarize this region past threshold, sodium channels open, and the influx of sodium ions makes the inside more positive. That positivity spreads to adjacent regions of the axon, nudging their channels to open in turn. The result is a chain reaction. Each segment of the membrane activates the next, sending the wave forward without loss of strength. One remarkable property of this process is its all-or-none nature. A small depolarization that does not reach threshold fades away, like a spark that fails to catch. But once threshold is crossed, the full event unfolds, no matter how much stronger the stimulus might have been. This guarantees reliability. Every action potential has essentially the same amplitude and duration, ensuring clarity and transmission. Intensity is encoded not in size but in frequency. Consider the sensation of heat on your skin. A faint warmth produces occasional action potentials in sensory neurons, while a stronger heat produces a rapid series. The brain interprets the frequency, not the amplitude, allowing it to distinguish between mild and intense stimulation. This principle applies across senses. Sound volume, light brightness, even the stretch of a muscle are all translated into firing frequency. The action potential is thus a universal currency, with rate as its unit of value. Conduction along the axon is influenced by structure. In unmyelinated axons, the wave moves continuously, each segment depolarizing the next in sequence. In myelinated axons, however, conduction takes a more efficient form called saltatory conduction. Here, insulating myelin wraps segments of the axon, leaving small gaps called nodes of Ranvier. Voltage-gated channels cluster at these nodes, so the action potential appears to jump from one node to the next. This leap greatly increases speed while conserving energy, since fewer ions need to be pumped back after each signal. The speed of conduction varies depending on axon diameter and degree of myelination. Large, heavily myelinated axons conduct quickly, suitable for reflexes that demand immediate response. Thin, unmyelinated fibers conduct slowly, often carrying information where urgency is less critical. The nervous system thus allocates resources according to function, ensuring both efficiency and adaptability. What prevents the wave from turning back on itself? The answer lies in the refractory period. After an action potential passes, sodium channels enter a temporary inactive state and cannot reopen immediately. This refractory phase enforces one-way travel, like a gate that closes behind each advancing spark. It also sets an upper limit on firing frequency, ensuring neurons cannot be driven beyond their safe capacity. The elegance of this system invites reflection. Though the action potential is purely electrical in nature, its consequences are vast. A wave traveling down a motor neuron may cause a muscle to contract, enabling movement. A train of action potentials in the auditory nerve allows the brain to reconstruct melody and speech. In the visual system, synchronized action potentials across networks create patterns that become images. The same event, repeated in countless variations, gives rise to perception, thought, and action. Yet the action potential is not only about communication, it also embodies balance. It arises from opposing forces, sodium rushing inward, potassium rushing outward, working in sequence to generate and then reset the signal. This interplay ensures that neurons are excitable, but not unstable, responsive but not chaotic. If the balance tilts, problems emerge. Excessive excitability can trigger seizures, while insufficient excitability can impair responsiveness. Thus, the action potential is both a symbol of vitality and a reminder of fragility. To appreciate its significance, imagine the brain without it. Each neuron would remain isolated, capable of local chemical changes, but unable to send messages across distance. Networks would collapse, coordination would vanish, and thought would never emerge. The action potential is therefore not just a mechanism, it is the enabling condition of consciousness itself. As this section draws to a close, picture again the traveling spark rising, spreading, resetting, and moving on. It is a rhythm repeated endlessly in your nervous system, even now as you listen and process these words. In the next section, we will explore what happens when that spark reaches the end of its journey, the synaptic bridges that transform electrical pulses into chemical signals between neurons. When an action potential reaches the end of an axon, it does not simply stop, it transforms. Electrical energy, having traveled down the membrane, must now cross a gap too wide for voltage alone. This is the synapse, a microscopic cleft that separates one neuron from another. Though the space is narrow, its presence forces a change in language. From electricity to chemistry, the synapse is thus both a barrier and a bridge, turning a fleeting spark into a molecular message. Inside the axon terminal, vesicles wait in readiness. Each vesicle is a tiny sphere filled with neurotransmitter molecules, packaged and prepared for release. At rest, they hover near the membrane, tethered but not yet fused. When the action potential arrives, calcium channels open, and calcium ions surge inward. This sudden rise in calcium is the signal for vesicles to act. Specialized proteins pull them into the membrane, where they merge and spill their contents into the synaptic cleft. In less than a heartbeat, chemistry takes over. The neurotransmitters drift across the gap, guided not by force but by diffusion. On the far side, the postsynaptic neuron awaits with receptors embedded in its membrane. These receptors are finely tuned, each recognizing a particular shape. When neurotransmitters bind, the receptors change form, opening ion channels, or triggering cascades within the cell. Depending on the transmitter and receptor type, the effect may excite the neuron toward firing, or inhibit it, holding activity in check. Excitatory synapses typically allow sodium or calcium to enter, depolarizing the postsynaptic cell. Inhibitory synapses often allow chloride to flow in or potassium to flow out, making the cell more negative and less likely to fire. The balance of these influences determines whether the neuron will reach threshold and launch its own action potential. In this way, each neuron acts as an integrator, weighing thousands of inputs to decide on a single output. Neurotransmitters come in many forms. Glutamate is the most common excitatory messenger, while gamma-aminobutyric acid, or GABA, provides inhibition. Acetylcholine plays roles in both movement and cognition, while dopamine, serotonin, and norepinephrine modulate mood, motivation, and attention. These molecules do not act alone. Their effects depend on receptor type and distribution, making the same neurotransmitter capable of very different outcomes in different circuits. Synaptic transmission is fast, but it is also tightly controlled. Once released, neurotransmitters must not linger indefinitely. Some are taken back up into the presynaptic terminal through transporter proteins, ready to be repackaged. Others are broken down by enzymes into inactive parts. This clearance ensures that signals remain brief, allowing rapid cycles of communication without blurring one into the next. The adaptability of synapses is one of their most remarkable features. Repeated activity can strengthen them, making neurotransmitter release more likely or receptor response more potent. This strengthening underlies learning and memory, allowing experiences to leave lasting traces in neural circuits. Conversely, unused synapses may weaken, conserving resources and keeping circuits efficient. This plasticity gives the brain its ability to change without losing stability. One might think of synapses as tiny marketplaces. Vesicles deliver goods across the divide, receptors receive them, and the flow of supply and demand determines the outcome. But unlike markets, the exchange is measured in milliseconds and repeated endlessly. In every moment, trillions of such transactions are occurring within your brain, sustaining thought, movement, and awareness. The existence of chemical synapses invites reflection on why such complexity is necessary. Why not allow neurons to connect directly, letting current flow without mediation? In fact, such direct connections do exist, called electrical synapses, where ions pass through gap junctions between cells. These allow rapid, synchronous firing, useful in reflexes and escape behaviors. But chemical synapses provide flexibility. Signals can be amplified, inhibited, modulated, and reshaped. They allow experience to alter future responses, something pure electrical continuity could not achieve. As this section closes, consider the synapse as a point of transformation. It is where the electrical language of action potentials becomes the chemical language of neurotransmitters, and then returns again to electricity in the postsynaptic cell. This constant translation is what makes the nervous system versatile, able to adapt and to learn. In the next section, we will explore the balance between excitation and inhibition, a dance that stabilizes the entire network. Every signal in the brain must pass through a gate of balance. On one side lies excitation, the push that drives neurons toward firing. On the other side lies inhibition, the pull that restrains them. The two forces coexist in every circuit, shaping not only whether a neuron fires but how information flows through networks. This balance is not fixed but dynamic, shifting moment by moment as needs change. Without it, the nervous system could not function with stability or precision. Excitatory transmission is dominated by glutamate, the most abundant neurotransmitter in the brain. When released at synapses, it binds to receptors that allow sodium and calcium to flow in, depolarizing the postsynaptic cell. This depolarization increases the likelihood that the neuron will reach threshold and fire its own action potential. Excitation spreads activity forward, ensuring that signals are not lost but carried on through circuits. In contrast, inhibition relies largely on gamma-aminobutyric acid, or GABA. When GABA binds to its receptors, channels open for chloride or potassium, making the inside of the neuron more negative. This hyperpolarization moves the cell further from threshold, decreasing its chance of firing. Inhibition acts as a counterweight, limiting runaway excitation and shaping patterns of activity. Rather than silence, it provides refinement, like a sculptor chiseling away excess stone to reveal form. The interplay of excitation and inhibition is evident in sensory systems. In the visual cortex, excitatory signals pass information about light, while inhibitory inputs sharpen edges and contrasts. This process, called lateral inhibition, makes borders appear clearer, allowing you to distinguish objects in a scene. Without inhibition, vision would blur, as light from neighboring regions overlapped without refinement. With inhibition, perception becomes precise. In motor systems, inhibition prevents opposing muscles from contracting simultaneously. When you extend your arm, excitatory signals activate the extensor muscles, while inhibitory signals suppress the flexors. This coordination allows smooth, efficient movement. A failure of inhibition here would result in stiffness or spasms, as both sets of muscles pulled at once. At a larger scale, Excitation and inhibition contribute to rhythms of activity across the brain. Networks of excitatory pyramidal cells and inhibitory interneurons generate oscillations that synchronize distant regions. These rhythms support functions from attention to sleep, binding together areas that must cooperate. Inhibition does not simply dampen activity, it helps set the tempo of neural communication. The ratio of excitation to inhibition is tightly regulated. Too much excitation can lead to seizures, where uncontrolled firing spreads through networks. Too much inhibition can suppress responsiveness, as seen in states of sedation or certain disorders of consciousness. The brain maintains balance through feedback loops, receptor regulation, and plastic changes at synapses. This dynamic equilibrium is sometimes called the EI balance, a central principle of neuroscience. Inhibitory neurons themselves show remarkable diversity. Some act locally, controlling small clusters of cells, while others project more broadly. Fast-spiking interneurons can synchronize large populations, ensuring that activity occurs in bursts rather than chaos. Others target dendrites, soma, or axon initial segments, each with a distinct role in modulating excitatory drive. This specialization makes inhibition versatile, able to shape signals in multiple ways. Excitation also carries layers of complexity. Different glutamate receptors, such as AMPA and NMDA types, contribute to both fast transmission and longer-term plasticity. The NMDA receptor, in particular, requires both depolarization and glutamate binding to open, acting as a coincidence detector. This property allows it to play a key role in learning, strengthening synapses when activity is coordinated. Thus, even within excitation, subtle mechanisms ensure that not every signal is treated equally. One can imagine excitation and inhibition as opposing currents in a river. Excitation pushes the water forward, giving force and direction. Inhibition guides the flow, preventing floods and carving channels. Together they shape the river into a pathway that sustains life downstream. Without either, the system would fail, either dried into silence or overwhelmed in chaos. As this section winds down, reflect on the quiet power of inhibition. Though less celebrated than excitation, it is essential for clarity, timing, and safety in every circuit. In the next section, we will meet the often overlooked partners of neurons, the glial cells, whose support and regulation make this balance possible. For many years, neuroscience treated glial cells as little more than scaffolding, a silent background against which neurons performed the true work. The very word glia comes from the Greek for glue, reflecting this belief. Yet as research advanced, a new picture emerged. Glial cells are not passive supports but active participants in brain function. They guide growth, maintain balance, insulate axons, and even influence signaling at synapses. Without them, neurons could not survive, let alone coordinate into circuits. There are several main types of glial cells, each with its own roles. Astrocytes, named for their star-like shape, weave among neurons and blood vessels, forming a vast supportive network. They regulate the concentration of ions and neurotransmitters in the extracellular space, preventing toxic buildup. They also supply neurons with energy substrates and help repair tissue after injury. Astrocytes extend fine processes that can wrap around synapses, influencing transmission directly. In doing so, they help determine when a signal should be amplified and when it should fade. Another key group are oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. These cells create myelin, the insulating sheath that wraps around axons. Myelin allows action potentials to travel rapidly by saltatory conduction, leaping from node to node rather than creeping along continuously. In this way, glial cells enable neurons to communicate across long distances with speed and efficiency. Damage to myelin, as seen in certain disorders, slows or disrupts conduction, leading to weakness, sensory changes, and fatigue. Microglia serve as the brain's immune sentinels. Unlike neurons and other glia, they originate from immune lineage and retain the ability to patrol, detect, and respond to threats. When injury or infection occurs, microglia change shape, migrate to the site, and release molecules that coordinate defense and repair. They also prune synapses during development and in adulthood, sculpting circuits by removing connections that are weak or unnecessary. This refinement helps maintain efficiency and prevents overcrowding of signals. Ependymal cells line the ventricles of the brain and the central canal of the spinal cord. They help produce and circulate cerebrospinal fluid, which cushions the nervous system and provides a medium for nutrient exchange. Though less discussed, they form part of the protective environment that keeps neurons stable. Together, These glial populations create a hidden infrastructure. They regulate the chemical environment, provide energy, remove waste, insulate connections, defend against pathogens, and shape circuits. It is no exaggeration to say that glia are the caretakers of the nervous system, enabling neurons to dedicate themselves to signaling. Recent discoveries suggest that glia also play active roles in communication. Astrocytes, for example, can release signaling molecules of their own, sometimes called gliotransmitters. These can modulate synaptic strength, synchronizing groups of neurons or influencing plasticity. Microglia, by pruning synapses, alter the very architecture of networks. Even oligodendrocytes may adjust myelination in response to activity, fine-tuning conduction speed to optimize coordination. The partnership between neurons and glia is not always smooth. When glia overreact, inflammation can spread, contributing to damage in conditions such as neurodegenerative diseases. When they fail, the consequences can be equally severe, as neurons lose the support they require. Understanding glia has become one of the most active frontiers in neuroscience, revealing layers of complexity once overlooked. To appreciate their significance, imagine a city at night. The lights and movement may suggest that only the visible traffic matters, but beneath lies a hidden network: roads, utilities, sanitation, and emergency services. Without these, the city would collapse within hours. Neurons are like the traffic, carrying visible signals. Glia are the infrastructure, unseen but indispensable, keeping everything flowing. As we end this section, consider that every thought, memory, and dream depends not only on neurons, but also on this silent majority of glial cells. They are the quiet partners that sustain life and learning. In the next section, we will see how individual neurons and their glial companions join into small circuits, the motifs from which all networks are built. When two neurons connect, the result is simple: one excites or inhibits the other. But when three or more are linked, patterns begin to emerge. These small arrangements of neurons, called microcircuits or motifs, are the building blocks of larger networks. Just as a composer uses recurring themes to structure a symphony, the brain reuses motifs across regions, applying them to vision, movement, memory, and emotion. One of the simplest motifs is feed-forward excitation. Here, a presynaptic neuron activates a second, which in turn activates a third. This chain allows information to pass rapidly, preserving the timing of the original signal. In reflex pathways, feed-forward excitation ensures that sensory input leads to swift motor response, such as withdrawing a hand from a hot surface. A variation is feed-forward inhibition. In this arrangement, an excitatory neuron activates both a target cell and an inhibitory interneuron. The interneuron then suppresses the target. The effect may seem counterintuitive, but it provides precision, sharpening the timing of responses. The target cell fires only briefly before inhibition arrives, preventing excessive or prolonged activity. Another recurring motif is feedback inhibition. Here, a neuron excites an interneuron, which then inhibits the original neuron. This loop acts as a brake, limiting activity once it begins. Feedback inhibition prevents runaway excitation, keeping circuits stable. It also allows rhythmic oscillations, as activity rises, is suppressed, and then rises again. Such rhythms appear in motor control and in cortical activity during attention and sleep. Lateral inhibition is another powerful motif. In this pattern, active neurons inhibit their neighbors. The result is contrast enhancement, sharpening the difference between signals. In the visual system, lateral inhibition makes edges stand out, allowing us to perceive borders with clarity. In sensory maps, it refines signals so that subtle differences are not lost. Convergence and divergence are structural motifs that shape flow. Convergence occurs when many inputs synapse onto a single neuron, allowing integration of information from multiple sources. Divergence is the opposite, one neuron projecting to many targets, amplifying its influence. Together, these arrangements allow the nervous system to combine detail with reach, ensuring both precision and breadth. More complex motifs include recurrent excitation, in which neurons loop back to activate each other. This creates sustained activity, even after the initial input ends. Such persistence is useful for holding information in working memory, or for maintaining attention on a task. But recurrent excitation must be balanced by inhibition, or it risks spiraling into instability. Microcircuits can also generate rhythms. When excitatory and inhibitory neurons are arranged in loops, they produce oscillations at different frequencies. These rhythms can synchronize across regions, coordinating activity. For example, Rhythmic circuits in the spinal cord generate patterns of movement for walking, while cortical rhythms contribute to perception and memory. Though motifs are small, their influence is immense. By combining them in countless variations, the brain creates circuits capable of everything from reflexes to abstract reasoning. The same motifs reappear in sensory systems, motor pathways, and cognitive networks, adapted to context. This reuse reflects evolutionary efficiency, a limited set of designs applied in diverse ways. To imagine their significance, think of language. A handful of letters combine into words, words into sentences, sentences into stories. Similarly, motifs combine into circuits, circuits into networks, networks into the living story of thought. The richness of the brain arises not from infinite variety, but from infinite recombination of simple patterns. As this section closes, hold the image of small loops and chains weaving together, forming the foundation of complexity. In the next section, we will explore a principle that gives these motifs lasting power, the strengthening of connections through coordinated activity, often summarized in the phrase cells that fire together, wire together. The phrase cells that fire together, wire together is one of the most enduring ideas in neuroscience. It captures the essence of Hebbian plasticity, the principle that connections between neurons strengthen when they are active at the same time. Though simple in wording, this rule provides a mechanism for learning, memory, and adaptation. It transforms fleeting patterns of activity into lasting changes in circuitry. At the core of this principle is the synapse. When a presynaptic neuron releases neurotransmitters and the postsynaptic cell responds strongly, molecular changes occur. Receptors may become more numerous, channels more responsive, or signaling pathways more efficient. If this pairing repeats, the synapse becomes more likely to transmit signals in the future. The connection is not fixed but modifiable, shaped by experience. This strengthening is often referred to as long-term potentiation, or LTP. It was first observed in the hippocampus, a region critical for memory. In experiments, brief but intense stimulation of synapses led to enhanced responses that lasted for hours, days, or longer. LTP depends on specific receptors, such as NMDA receptors, that act as coincidence detectors. They open only when presynaptic release and postsynaptic depolarization occur together. In this way, the synapse knows when two cells are active simultaneously and adjusts accordingly. The consequences of Hebbian plasticity extend beyond single synapses. When groups of neurons fire together, networks of strengthened connections emerge. These assemblies form the neural basis of association. If you see a particular object while hearing a certain sound, the neurons representing sight and sound may fire together, binding the two experiences. Later, encountering one may bring the other to mind. This is the foundation of associative memory. Hebbian principles also explain how skills are refined. Repeated practice strengthens the circuits that are consistently active, making movements smoother and more automatic. In sensory systems, repeated exposure can sharpen perception as circuits become tuned to relevant features. Plasticity allows the nervous system to adapt to new environments and demands, sculpting itself according to use. Yet plasticity is not limitless. Connections cannot strengthen indefinitely, or the system would lose balance. Mechanisms, such as long-term depression, the weakening of synapses with low or uncoordinated activity, provide counterweight. Homeostatic processes ensure that overall activity remains within functional bounds. In this way, the brain balances stability with flexibility, preserving both continuity and change. There is beauty in the efficiency of this rule. Rather than storing memories in a central location, the brain distributes them across networks of strengthened synapses. Each memory is a pattern of activity woven into circuits, resistant to loss, yet capable of modification. This distributed storage allows resilience. Even if some neurons are damaged, the memory may persist in the remaining pattern. The Hebbian idea also invites reflection on identity. If connections are shaped by repeated activity, then our experiences, choices, and habits literally sculpt the structure of our brains. The paths we walk most often become the ones most deeply carved. This perspective underscores both the adaptability and the responsibility of living with a nervous system that is always learning. At the same time, Hebbian plasticity has limits in speed and capacity. Not every coincidence of firing leads to change, and not all changes endure. The brain filters significance, favoring strong, repeated, or emotionally charged experiences. This selectivity prevents overload, allowing meaningful events to stand out. As this section closes, Hold in mind the image of two neurons firing together, their connection strengthening with each encounter. Multiply this across countless pairs, and you have a brain that remembers, adapts, and grows from experience. In the next section, we will examine the boundaries of this plasticity, considering both its possibilities and its constraints. Plasticity allows the brain to adapt, but it is not boundless. Every synapse has limits, every network boundaries that prevent endless growth. These constraints are as important as the capacity for change, for without them, circuits would lose stability, and memories would blur into noise. The nervous system balances flexibility with structure, creating a system that learns while preserving coherence. One limit lies in saturation. Synapses can strengthen only so far. Receptors and signaling pathways cannot expand indefinitely. Once a synapse reaches maximum efficacy, further activity yields little additional change. This ceiling ensures that no single connection overwhelms the network. It also preserves diversity, allowing multiple inputs to retain influence rather than being crowded out by one dominant source. Another boundary is time. Plastic changes occur more readily during certain developmental periods, often called critical windows. During these times, the brain is especially receptive to shaping by experience. Language acquisition in early childhood is one example, where exposure guides permanent wiring of speech and comprehension circuits. After the window closes, learning is still possible, but requires more effort, and some features may never be acquired with the same fluency. These sensitive periods highlight both opportunity and constraint. Plasticity also competes with stability. Too much change would destabilize memories and skills, erasing them with each new experience. To prevent this, the brain employs homeostatic mechanisms. If overall activity in a neuron becomes too high, synaptic strengths may scale down across the board. If activity drops too low, they may scale up. This global adjustment keeps circuits within functional ranges, much like a thermostat keeping temperature steady despite fluctuations. There are also structural limits. Neurons cannot form unlimited new connections. Space, energy, and resources impose boundaries. Growth of dendrites and synapses requires protein synthesis, energy, and coordination with glial support. Excessive connections could clog communication, slowing rather than enhancing function. Pruning ensures efficiency, trimming unused synapses so that networks remain streamlined. This balance between growth and elimination is evident throughout development and continues in adulthood. Plasticity carries risks when unchecked. In some disorders, excessive strengthening or inadequate pruning may contribute to dysfunction. Circuits may become too rigid, resisting adaptation, or too unstable, rewiring constantly without consolidation. Both extremes highlight the importance of balance. Adaptation is valuable, but it must occur within a framework that maintains reliability. Another constraint is context. Not every pairing of activity leads to learning. Attention, motivation, and emotional state influence plasticity. Neurochemical signals such as dopamine can tag experiences as significant, promoting synaptic change. In their absence, activity may fade without leaving lasting marks. This selectivity ensures that the brain does not encode trivial events with the same weight as important ones. Reflecting on these boundaries brings perspective. While it is tempting to view the brain as endlessly malleable, its constraints are what make meaningful change possible. Like clay that can be shaped only within certain limits, neural circuits are sculpted by both potential and restriction. Too much rigidity would leave us inflexible. Too much fluidity would erase continuity. The interplay creates a system that can learn while preserving identity. Plasticity's limits also remind us that not every habit is easily unlearned, not every skill readily mastered, and not every memory fully erasable. These constraints shape human experience, grounding us in both possibility and imperfection. They ensure that while we may grow and adapt, we also remain anchored, with a sense of continuity across time. As this section ends, imagine the brain as a garden. Growth occurs where conditions permit, but soil, sunlight, and boundaries of space define what is possible. The next section will turn to a different kind of shaping force: attention, the selective spotlight that guides which signals are amplified and which fade into the background. Attention is often compared to a spotlight, a narrow beam that illuminates certain parts of a scene while leaving the rest in shadow. In the brain, attention works in much the same way. Among the countless signals arriving from the senses, only a fraction can be processed in depth. Attention allows selection, amplification, and focus, guiding perception and action toward what matters most. At its foundation, attention is not a single process, but a network of interacting systems. Sensory areas filter inputs, while frontal regions set goals and priorities. Parietal regions contribute to spatial selection, helping determine where the spotlight should fall. Together, these systems coordinate the allocation of limited resources, ensuring that important stimuli are enhanced while distractions are suppressed. The effects of attention can be seen in the cortex. When you focus on an object, neurons in the relevant sensory area increase their responsiveness. For example, directing attention to a region of the visual field boosts activity in corresponding parts of the visual cortex. Signals become stronger, more reliable, and more likely to influence downstream processing. At the same time, unattended information weakens, fading from influence even though it still reaches the senses. Attention is guided by both bottom-up and top-down factors. Bottom-up attention responds to sudden or salient stimuli, such as a flash of light or a loud sound. It is fast, automatic, and protective, ensuring that potential threats or opportunities are not missed. Top-down attention, in contrast, is goal-driven. It reflects intentions, such as searching for a friend in a crowd or reading a page of text despite background noise. Both forms of attention interact, balancing responsiveness with control. The timing of attention reveals its dynamic nature. Shifts of focus can occur within fractions of a second, allowing rapid adaptation. Yet attention is also limited. It cannot fully process many items at once. This constraint forces selection, emphasizing some aspects of the environment while leaving others unattended. The spotlight can move, but it cannot illuminate everything simultaneously. Attention is not restricted to vision. Auditory attention allows you to follow one voice in a noisy room, filtering out competing sounds. Somatosensory attention lets you focus on the feel of an object in your hand while ignoring background sensations. Even internal signals, such as thoughts or emotions, can become the focus of attention, shaping experience and behavior. Neuromodulators such as acetylcholine and norepinephrine play key roles in attention. Acetylcholine enhances sensory responses, sharpening cortical processing. Norepinephrine adjusts responsiveness, increasing alertness to unexpected events. These chemical signals adjust the gain of neural circuits, tuning them for selective focus. Without them, attention would lack flexibility and precision. The limits of attention are evident in everyday life. Multitasking often feels efficient, but in truth it divides resources and reduces depth of processing. Shifting focus carries costs, as circuits must reconfigure. This is why sustained concentration can produce deeper learning than fragmented attention. The brain can switch tasks, but not without losing some fidelity in each. Attention also interacts with plasticity. Signals that receive focused attention are more likely to leave lasting traces in memory. By amplifying certain inputs, attention guides which synapses strengthen and which fade. In this sense, attention acts as a teacher, highlighting what the brain should encode. Without attention, learning is slower and less robust. Reflecting on attention reveals both its power and fragility. It allows us to select from the flood of information, shaping perception and memory. Yet it is limited, vulnerable to distraction, and easily fatigued. The balance between focus and openness, control and spontaneity, defines much of human experience. As this section closes, imagine a darkened theater where a single spotlight moves across the stage, illuminating actors one by one. The rest of the cast waits in shadow, unseen but present. In the next section, we will follow one of those actors, the visual system, and see how patterns of light are transformed into shapes, colors, and scenes within the brain. Vision begins with light, but it becomes perception only through the work of neural circuits. Photons entering the eye strike the retina, where specialized cells convert them into electrical signals. From that point onward, the brain must interpret patterns of activity as shapes, colors, and motion. What seems effortless, recognizing a face, reading a word, is the result of layers of processing that gradually transform simple inputs into complex representations. The retina is the first stage of this journey. Photoreceptor cells, rods and cones, absorb light and trigger changes in their membrane potential. Rods are highly sensitive and excel in dim conditions, while cones support color vision and fine detail. Their outputs pass through intermediate cells, including bipolar and horizontal cells, which integrate and refine the signals before sending them to ganglion cells. The axons of these ganglion cells bundle together to form the optic nerve, carrying visual information toward the brain. Already in the retina, processing is sophisticated. Lateral inhibition sharpens contrast, making edges more distinct. Some ganglion cells respond best to small spots of light, others to changes in motion or contrast. This early analysis prepares signals for further refinement in the brain. The optic nerve fibers travel first to the lateral geniculate nucleus of the thalamus, a relay center that organizes input before sending it to the visual cortex. In the primary visual cortex, located at the back of the brain, neurons respond to specific features. Some detect edges at particular angles, others respond to motion in certain directions. By combining these responses, the cortex constructs a map of the visual field, with different areas devoted to different aspects of the scene. Higher-order regions then build upon this foundation, integrating color, depth, and shape into coherent objects. Color perception relies on comparisons among different types of cones, each sensitive to particular wavelengths of light. The brain interprets relative activity across these cones to generate the experience of color. Depth perception emerges from binocular disparity, the slight difference between images captured by the two eyes. Motion detection arises from circuits tuned to changes in position over time. Each of these features depends on networks of neurons specialized for distinct tasks. The visual system is organized hierarchically. Lower levels process basic features, while higher levels combine them into complex representations. In some cortical areas, neurons respond specifically to faces, places, or objects. This specialization allows rapid recognition, essential for survival and social interaction. Yet the system remains flexible, adapting to changes in input and experience. For example, if visual input is altered, circuits can adjust their responses, reflecting the plasticity of the brain. Attention interacts strongly with vision. When you focus on a particular region of a scene, activity in the corresponding cortical area increases, enhancing detail and clarity. Unattended regions fade, even though they are still represented. This selective amplification helps manage the immense amount of information arriving at the eyes every moment. The richness of vision also comes with vulnerabilities. Optical illusions exploit the brain's assumptions, revealing how perception can diverge from reality. Blind spots created where the optic nerve exits the retina are usually unnoticed because the brain fills them in with surrounding information. These phenomena highlight that vision is not a passive recording but an active construction, shaped by both input and expectation. Reflecting on vision invites awe. From the landing of photons on the retina to the recognition of a familiar face, countless steps occur in fractions of a second. Circuits work in parallel, integrating features seamlessly into experience. The simplicity of sight conceals the complexity of its mechanisms. As this section closes, imagine light striking the eye and being woven into a vivid image by the brain. In the next section, we will turn from sight to sound, following vibrations of air as they become signals that carry tone, pitch, and meaning. Hearing begins not with electricity but with movement, vibrations in the air that press against the ear. These vibrations, carried as waves of pressure, enter the outer ear and travel down the canal to strike the eardrum. The eardrum responds by oscillating back and forth, setting in motion a chain of tiny bones in the middle ear. These bones, the malleus, incus, and stapes, amplify the vibrations and deliver them to the inner ear, where mechanical motion is transformed into neural signals. The inner ear contains the cochlea, a spiraled structure filled with fluid. Running along its length is the basilar membrane, which responds differently depending on frequency. High-frequency sounds cause movement near the base, while low-frequency sounds resonate near the apex. This spatial organization creates a tonotopic map, where each region of the membrane is tuned to a specific pitch. The cochlea thus decomposes complex sounds into their frequency components before they even reach the brain. On the basilar membrane sit rows of hair cells, the true sensory receptors of hearing. Each hair cell carries bundles of tiny projections called stereocilia. When the basilar membrane vibrates, the stereocilia bend, opening ion channels that allow potassium and calcium to flow in. This depolarization leads to the release of neurotransmitters at the base of the hair cell, activating auditory nerve fibers. In this way, mechanical energy is converted into electrical signaling. The auditory nerve carries this information into the brainstem, where it is processed in several nuclei. Some circuits measure the time difference between sounds reaching the two ears, allowing localization of direction. Others compare intensity across ears, refining spatial perception. Signals then ascend through the midbrain and thalamus before reaching the auditory cortex, located in the temporal lobe. In the cortex, neurons are arranged according to frequency, preserving the tonotopic map established in the cochlea. This organization allows precise representation of pitch. Beyond pitch, cortical circuits analyze patterns over time, extracting rhythm, melody, and speech. Specialized regions respond selectively to language, enabling comprehension of words and sentences. Other areas are attuned to music, revealing emotional and structural aspects of sound. Auditory processing is not only bottom-up but also top-down. Expectations, attention, and experience shape what we hear. In a noisy environment, focusing on one voice enhances its representation while suppressing background chatter, a phenomenon often called the cocktail party effect. This selective processing reflects the interaction of sensory input with cortical control. The auditory system also shows remarkable plasticity. With training, the brain can refine its representation of subtle differences in pitch or timing. Musicians often display enhanced responses in auditory cortex, reflecting years of practice. Similarly, language learning can sharpen distinctions between phonemes that are absent in one's native tongue. These adaptations underscore the flexibility of auditory circuits. Yet the system has vulnerabilities. Damage to hair cells, which do not regenerate in humans, can lead to permanent hearing loss. Excessive noise exposure, aging, or genetic factors can impair function. The brain can compensate to some degree, enhancing sensitivity of remaining inputs, but compensation has limits. This fragility reminds us of the delicacy of the mechanisms underlying sound. Reflecting on hearing, one sees parallels to vision. Just as the visual system decomposes light into features, the auditory system decomposes sound into frequency, timing, and intensity. Both rely on hierarchical processing, building complexity from simple inputs. Both are shaped by attention and expectation, turning raw signals into meaningful experience. As this section closes, imagine a simple vibration in the air, perhaps the sound of a single note. Within your ear and brain, it is unraveled into pitch, tone, and harmony, then reassembled into the experience of music or speech. In the next section, we will move from sound to touch, exploring how the body maps itself in the brain through patterns of sensation. Touch is our most immediate link to the physical world. A gentle brush, the weight of an object in the hand, the sharpness of a pinprick, all of these arise from receptors embedded in the skin and tissues. These receptors detect mechanical, thermal, and painful stimuli, converting them into electrical signals that travel to the brain. From there, perception emerges not as a raw stream, but as an organized map of the body. Different receptors specialize in different aspects of sensation. Some respond best to light pressure and fine detail, others to vibration or stretch. Thermoreceptors sense warmth and coolness, while nociceptors respond to potentially harmful stimuli. Each type of receptor connects to sensory neurons that carry information through peripheral nerves into the spinal cord. From there, pathways ascend to the brainstem and thalamus before reaching the somatosensory cortex. In the cortex, touch is represented in a strikingly ordered fashion. Neighboring regions of the body project to neighboring regions of the cortex, creating a somatopic map. This map, sometimes called the homunculus, depicts a distorted human figure with large hands, lips, and tongue, reflecting the high density of receptors in those areas. The map is not fixed but plastic, capable of reorganizing when input changes. For example, if a person practices a skill requiring fine finger movements, the cortical representation of the fingers can expand. Conversely, loss of input from a limb may lead to neighboring regions taking over its cortical space. This adaptability highlights the dynamic nature of sensory maps, always adjusting to experience and use. Touch is not only about location, but also about integration. Signals from different receptors combine to create complex perceptions. Texture, for instance, is sensed through a mixture of fine detail from slow-adapting receptors and vibration from fast-adapting ones. The brain merges these inputs, producing a coherent sense of surface quality. Similarly, weight and shape are judged by combining pressure and proprioceptive signals from muscles and joints. Inhibition plays a key role in refining touch. As in vision, lateral inhibition sharpens contrast, making borders more distinct. This allows you to distinguish the edges of an object with precision. Without inhibition, touch would blur, and surfaces would feel less defined. The clarity of tactile perception depends on this interplay of excitation and suppression. Pain is part of the somatosensory system, though it is distinct in many ways. Nociceptors respond to damaging or threatening stimuli, sending signals that warn of harm. Pain perception is shaped not only by sensory input but also by context, emotion, and attention. The same stimulus may feel more or less painful depending on expectation or focus. This modulation reflects the brain's active role in shaping sensation. The sense of touch also extends inward, informing us of the position and movement of our bodies. Proprioceptors in muscles, tendons, and joints provide continuous feedback about posture and motion. These signals allow coordination, balance, and fine motor control. They are integrated with tactile input to create a seamless sense of bodily presence. Reflecting on touch reveals its intimacy. Unlike vision or hearing, which perceive at a distance, touch requires direct contact. It grounds us in the here and now, anchoring perception to the physical body. Through touch, we explore the environment, learn about objects, and connect with others. As this section closes, imagine running your hand over a textured surface. Beneath that simple act, countless receptors fire, signals ascend through pathways, maps activate in cortex, and perception emerges. In the next section, we will follow another ancient sense, smell, and see how its pathways link directly to memory and emotion. Smell is often described as the most ancient of senses. Long before eyes and ears evolved, organisms navigated their environments through chemical detection. In humans, olfaction still carries this primal quality. A single scent can summon vivid memories or stir emotions, linking present experience to the past in a way that feels immediate and powerful. The journey of smell begins in the nasal cavity. In its upper region lies the olfactory epithelium, a sheet of specialized receptor neurons. Each of these neurons expresses a receptor tuned to particular molecular features. When odor molecules bind, the receptor changes shape, triggering an electrical response. Unlike many sensory neurons, these olfactory cells send their axons directly into the brain, converging on structures called glomeruli within the olfactory bulb. The olfactory bulb is organized in a precise map. Neurons carrying the same receptor type converge onto the same glomerulus, pooling their signals. This arrangement transforms the vast diversity of chemical molecules into spatial patterns of activity. Each odor produces a unique combination of glomeruli activation, like a fingerprint for the nose. From here, signals are relayed deeper into the brain. One striking feature of olfaction is its direct connection to regions involved in emotion and memory. Fibers from the olfactory bulb project to the amygdala and hippocampus without passing first through the thalamus, unlike other senses. This anatomical shortcut helps explain why smells evoke emotions and memories so vividly. The scent of pine may call forth winter mornings. The aroma of bread may recall family gatherings. These associations are not learned in textbooks but inscribed directly through experience. The piriform cortex, part of the olfactory system, contributes to recognizing and categorizing odors. Unlike the orderly maps of vision or hearing, olfactory representations are more distributed, with patterns of activity spread across regions. This distributed coding allows flexibility, enabling recognition of countless combinations of odor molecules. It also supports learning, as new experiences can reshape the associations. Smell is closely tied to taste, forming the combined perception of flavor. While taste buds detect basic qualities such as sweet, sour, salty, bitter, and umami, much of what we experience as flavor arises from olfactory input. This is why food seems bland when the nose is congested. The integration of taste and smell demonstrates how senses converge to enrich perception. Olfaction also contributes to behavior in subtle ways. Pheromones and other social cues, though less pronounced in humans than in some animals, may influence attraction and communication. Environmental odors can signal danger, such as smoke or spoilage, prompting rapid responses. In this way, smell remains a guide for survival, even in modern contexts. The system is not static. Olfactory neurons are among the few in the nervous system that regenerate throughout life. This turnover allows resilience, maintaining sensitivity despite exposure and damage. However, loss of smell can occur with aging or illness, sometimes preceding other neurological changes. Such vulnerability underscores the connection between olfaction and broader brain health. Reflecting on smell reveals its dual nature, both primal and deeply personal. It connects us to the environment through chemistry and to ourselves through memory. It bypasses analysis to reach directly into feeling, reminding us of the closeness of body and mind. As this section closes, imagine inhaling the aroma of a familiar place. In that moment, molecules drifting in the air spark activity that awakens memory and emotion. In the next section, we will move from the sensory world to the rhythmic patterns of the brain itself, exploring how oscillations shape rest, attention, and coordination. The brain is never still. Even in silence, even in sleep, waves of activity ripple through its networks. These rhythms, known as oscillations, are patterns of synchronized firing across groups of neurons. They vary in speed and function, from slow waves that dominate deep rest to faster rhythms that accompany attention and thought. Like the steady beat of a drum in an ensemble, oscillations provide timing that organizes the orchestra of neural activity. At the cellular level, oscillations arise from interactions between excitatory and inhibitory neurons. Excitatory cells drive activity forward, while inhibitory interneurons impose pauses, creating cycles of rise and fall. When many neurons align their activity, these cycles become detectable as brainwaves. The coordination is not imposed from outside, but emerges naturally from the balance of forces within circuits. Different rhythms serve different roles. Slow oscillations, prominent during deep sleep, help consolidate memories by replaying patterns of activity. Slightly faster rhythms, often linked to drowsiness or relaxation, create a bridge between waking and sleep. In the waking state, intermediate rhythms support attention and working memory, allowing networks in different brain regions to communicate efficiently. Faster oscillations emerge during focused thought and perception, binding together features into coherent experience. These rhythms are not confined to one region. They can synchronize across distant areas, creating functional networks. For example, oscillations may align between the visual cortex and frontal regions when attention is directed to a specific object. This alignment allows efficient communication, as spikes arrive at the optimal phase to influence downstream neurons. Without timing, signals might miss their targets or interfere with one another. Oscillations are flexible, shifting with state and demand. During rest, they slow, allowing recovery and integration. During active engagement, they accelerate, supporting rapid processing. The brain transitions smoothly between these modes, adjusting rhythm to context. This adaptability ensures both efficiency and resilience, preventing circuits from becoming locked in one pattern. Rhythms also provide windows into brain health. Disruptions in oscillatory patterns can accompany disorders of perception, memory, and consciousness. Too little synchronization may impair communication, while too much may lead to rigidity or seizures. The study of brain rhythms has therefore become central to understanding both function and dysfunction. Reflecting on oscillations reveals their subtlety. They are not thoughts themselves but the background structure that shapes thought. Just as music requires both melody and rhythm, neural activity requires both spikes and oscillations. The melody carries content, the rhythm organizes it into meaningful patterns. These rhythms also offer a sense of continuity. While individual neurons fire briefly, oscillations persist, linking moments into flows. They provide a temporal framework within which memories can be replayed, sensations integrated, and rest restored. In this way, rhythms are both physiological and experiential, shaping not only the efficiency of circuits, but also the felt quality of states like calm or alertness. As this section closes, imagine the brain as a sea, waves rising and falling, overlapping and merging. Each wave has its own pace, but together they form a dynamic surface that reflects the balance of excitation and inhibition beneath. In the next section, we will explore how one of the deepest rhythms, the circadian cycle, aligns brain and body with the cycle of day and night. Beneath the daily rhythms of thought and activity lies a larger rhythm, one that stretches across hours rather than seconds. This is the circadian cycle, the internal clock that aligns body and brain with the cycle of day and night. It regulates sleep and wakefulness, but also temperature, hormone release, metabolism, and mood. The circadian system ensures that the body's functions occur at the right times, coordinating physiology with the environment. The master clock resides in a small region of the hypothalamus, called the suprachiasmatic nucleus, or SCN. Though tiny, it contains thousands of neurons that keep time through molecular feedback loops. Genes within these cells produce proteins that accumulate, inhibit their own expression, degrade, and then restart the cycle. The rhythm repeats roughly every 24 hours, providing a biological basis for the day. These oscillations are remarkably stable, but can be reset by external cues. Light is the most powerful cue. Specialized photoreceptors in the retina detect brightness and send signals directly to the SCN. Morning light shifts the clock earlier, while evening light delays it. This entrainment ensures that internal time remains aligned with the external world. Without such cues, circadian rhythms would drift, running slightly longer or shorter than the solar day. The SCN coordinates rhythms across the body. It sends signals to other brain regions and influences hormonal release. One pathway regulates melatonin secretion from the pineal gland, which rises in darkness and falls with light. Another adjusts cortisol levels, promoting alertness in the morning and rest at night. Body temperature, digestion, and immune function all follow circadian patterns, showing how deeply the clock shapes physiology. Yet the system is not rigid. It can adjust to changes, though not instantly. Shifts in light exposure, such as during travel across time zones, require gradual realignment. Until the clock catches up, sleep and alertness may feel misaligned, producing the familiar discomfort of jet lag. Similarly, irregular schedules or exposure to artificial light at night can desynchronize rhythms, affecting mood, performance, and overall balance. Circadian rhythms also interact with learning and memory. Certain phases of the cycle are more favorable for encoding new information, while others support consolidation during rest. These time-dependent effects suggest that cognition is not uniform across the day, but shaped by the clock's influence on neural circuits. The circadian system demonstrates how molecular mechanisms scale up to whole-body effects. From feedback loops in single neurons of the SCN emerges a rhythm that orchestrates sleep, metabolism, and mood. This hierarchy, from gene expression to behavior, exemplifies the layered nature of neuroscience. Reflecting on circadian rhythms invites appreciation of their subtle guidance. They operate quietly, often unnoticed, yet they influence every aspect of daily life. When aligned with the environment, they provide stability and efficiency. When disrupted, they remind us of their importance by leaving us fatigued, restless, or unfocused. As this section closes, imagine the body as a vast orchestra, and the SCN as its conductor, ensuring that each section enters at the right time. In the next section, we will turn to circuits that shape emotion, exploring how regions such as the amygdala and prefrontal cortex generate feelings of fear, joy, and calm. Emotions arise from the coordinated activity of circuits that link perception, memory, and bodily state. Far from being vague feelings, they are structured patterns generated by specific neural systems. Among these, the amygdala and prefrontal cortex play central roles, working together to evaluate situations, assign value, and guide responses. Through these circuits, a simple stimulus, a sudden sound, a familiar face, can spark fear, joy, or calm. The amygdala, an almond-shaped cluster deep within the temporal lobe, is often described as a hub for emotional salience. It detects signals that may indicate threat or reward, rapidly preparing the body for action. When a potential danger appears, the amygdala activates autonomic responses, accelerated heart rate, heightened vigilance, release of stress hormones, long before conscious appraisal. This speed ensures survival, allowing organisms to react swiftly to possible risks. The amygdala achieves this through its widespread connections. Inputs arrive from sensory pathways, providing quick access to visual, auditory, and somatic cues. Outputs project to the hypothalamus and brainstem, triggering physiological responses, and to cortical regions, influencing perception and memory. In this way, the amygdala acts as a hub that links sensation, bodily state, and cognition into a unified reaction. Yet the amygdala does more than detect danger. It also encodes associations between stimuli and outcomes. If a neutral sound repeatedly precedes a startling event, the amygdala learns to respond to the sound itself. This form of conditioning demonstrates how emotional circuits shape future behavior, linking memory to survival. Once such an association is formed, it can guide behavior automatically, often without conscious recall of the original event. Balancing this rapid reactivity is the prefrontal cortex, particularly regions in the ventromedial and dorsolateral areas. The prefrontal cortex evaluates context, weighing long-term goals against immediate impulses. It can dampen amygdala responses when a threat proves harmless, or enhance them when vigilance is warranted. This dialogue allows emotions to be flexible rather than fixed, adjusting to circumstance. The dorsolateral prefrontal cortex supports deliberate regulation strategies, such as reappraisal. When a person reframes a stressful event as a challenge rather than a threat, activity in this region increases, correlating with reduced amygdala responses. The ventromedial prefrontal cortex, in contrast, plays a role in valuation, integrating emotional signals with expectations of future outcomes. Its connections to the amygdala allow it to fine-tune emotional intensity based on context and goals. The prefrontal cortex also interacts with memory. It draws on hippocampal input to assess whether a current stimulus matches a dangerous past situation or is benign in the present. This capacity prevents overgeneralization, such as responding with fear to all loud sounds rather than only those associated with real threats. When prefrontal regulation fails, emotions can become excessive or inappropriate, contributing to anxiety or impulsivity. Other regions contribute as well. The hippocampus provides contextual memory, helping distinguish between safe and dangerous environments. A snake encountered in the wild may trigger fear, but the same snake behind glass in a museum may not. The hippocampus supplies the contextual information that allows such discrimination. The insula represents internal bodily states, linking emotion to visceral sensations. Activity in the insula corresponds to awareness of heart rate, gut feelings, and other interoceptive cues. This integration gives emotions their bodily texture, the flutter of nervousness, the warmth of affection. Without insula input, emotions might lack their embodied quality, feeling abstract rather than lived. The anterior cingulate integrates conflict and control, guiding adjustments when emotion and task demand collide. It detects when automatic emotional reactions clash with goals, signaling the need for regulation. For example, anger may arise during a disagreement, but the anterior cingulate can help redirect behavior toward constructive dialogue rather than escalation. Together, these structures form a distributed network that generates and regulates emotional life. No single region creates emotion alone. Rather, emotions emerge from dynamic interactions among sensory, limbic, and cortical systems. The chemistry of emotion involves multiple neurotransmitters and modulators. Dopamine contributes to reward and motivation, reinforcing behaviors that lead to positive outcomes. Serotonin supports mood stability, modulating reactivity and resilience. Norepinephrine promotes arousal and alertness, amplifying signals of novelty or threat. These chemicals do not act in isolation, but adjust the sensitivity of circuits. Elevated dopamine activity may heighten reward sensitivity, making pleasurable experiences more salient. Reduced serotonin may increase vulnerability to mood swings. Altered norepinephrine signaling may amplify anxiety by exaggerating responses to minor stimuli. Thus, neurochemistry sets the tone for emotional experience, tuning circuits towards stability or volatility. Hormones also play a role. Cortisol, released during stress, feeds back to brain regions, such as the hippocampus and prefrontal cortex, influencing memory and regulation. Oxytocin, often associated with bonding, modulates amygdala activity and enhances trust. These modulators show how body and brain interact, linking endocrine states to neural circuits of emotion. Emotion circuits do not act in isolation. They interact with sensory and motor systems. A fearful stimulus heightens attention to the environment, narrowing focus on potential threats. A joyful one broadens exploratory behavior, encouraging openness to new ideas and actions. Emotional states bias perception, memory, and decision-making, coloring experience in ways both subtle and profound. This integration shows that emotion is not an addition to cognition, but an inseparable partner. The traditional view of reason versus emotion is misleading. In truth, cognition is always infused with emotional tone, and emotion is shaped by cognitive appraisal. The brain evolved not to separate them but to unite them, ensuring that thought is guided by value and that feeling is informed by context. Emotions also carry adaptive value. Fear promotes avoidance of harm, mobilizing flight or defensive behaviors. Anger energizes confrontation, mobilizing resources for defense or boundary setting. Joy reinforces beneficial behavior, encouraging repetition of rewarding activities. Sadness signals the need for rest or support, fostering social connection. Disgust protects against contamination, steering organisms away from harmful substances. By shaping behavior in ways that enhance survival and social bonds, emotions reveal their evolutionary roots. They are not arbitrary states but strategies honed by natural selection, efficient shortcuts for guiding action in complex environments. At the same time, these circuits are not infallible. They can overreact, producing anxiety in safe settings, or underreact, dulling responses to genuine threats. When fear generalizes too broadly, vigilance becomes chronic anxiety. When regulation fails, sadness may deepen into depression. Overactivation of reward circuits may contribute to addiction, where stimuli acquire excessive motivational pull. Regulation by prefrontal and contextual systems is essential to maintain balance. The interplay between rapid amygdala responses and slower cortical evaluation defines much of human emotional life. When the balance tips too far toward reactivity, emotions overwhelm. When it tips too far toward suppression, emotions lose vitality. Adaptive function lies in the middle, where emotion informs but does not dominate. Emotions are also shaped by culture and social context. While basic circuits are universal, their expression and regulation are influenced by norms and learning. The same neural circuitry that generates fear or joy is modulated by cultural practices, expectations, and values. For example, anger may be encouraged as assertiveness in one setting and discouraged as disruption in another. Social interaction further amplifies emotions through mirroring. The sight of another person's expression can trigger similar patterns in the observer's brain, aligning states and fostering empathy. This resonance demonstrates how emotions are not purely individual but relational, linking nervous systems into shared experience. Reflecting on these mechanisms reveals the depth of emotional experience. Fear is not simply a feeling, but the coordinated firing of amygdala neurons, release of stress hormones, tightening of muscles, and sharpening of attention. Joy involves activation of reward pathways, relaxation of physiological tension, and expansion of thought. Each state is a whole-body phenomenon, orchestrated by neural circuits. Emotions are thus bridges between body and brain, past and present, self and others. They inform decisions, enrich memory, and guide social bonds. Their complexity reflects the integration of circuits across levels, from molecules to networks, from reflexes to reflection. As this section closes, imagine the amygdala as a sentinel, always scanning for significance, and the prefrontal cortex as a judge, weighing evidence before allowing action. Around them, the hippocampus, insula, and cingulate provide context, sensation, and adjustment. The orchestra plays not in isolation but in harmony, generating the symphony of emotional life. In the next section, we will explore how memory systems record experience, linking the hippocampus and cortex in the creation of lasting knowledge. Memory is not a single function, but a family of processes, each supported by distinct yet interconnected circuits. Experiences must be encoded, stored, and later retrieved, and different forms of memory rely on different brain systems. The hippocampus and cortex, in particular, form a partnership that allows fleeting events to become lasting knowledge. Together, they preserve the threads of personal history and weave them into the fabric of identity. The hippocampus, located deep within the medial temporal lobe, is often described as a gateway to memory. It receives highly processed input from sensory areas and binds together the components of an experience, sights, sounds, emotions, and spatial context, into a unified representation. This process, known as encoding, ensures that memories are not isolated fragments, but coherent episodes. The hippocampus is especially important for declarative memory, the kind that allows one to recall facts and events. Within the hippocampus, specialized circuits contribute to its function. Place cells, for example, fire when an individual is in a specific location, forming a cognitive map of space. Grid cells found in neighboring entorhinal cortex create a coordinate system, helping track position even without external cues. These spatial representations are integrated with sensory and emotional information, allowing experiences to be grounded in context. A walk through a childhood home may awaken not just a visual memory of the rooms but also the emotional tone tied to them, a direct result of hippocampal integration. After encoding, memories are not stored permanently in the hippocampus. Instead, they are gradually transferred to the neocortex, a process known as consolidation. During sleep, particularly in slow-wave stages, hippocampal activity replays patterns of firing from recent experiences. This replay strengthens connections in cortical circuits, embedding the memory more permanently. The cortex, with its vast capacity, acts as a long-term archive, integrating memories into networks of related knowledge. Over time, the hippocampus becomes less critical for retrieval, as the cortex can reconstruct the memory independently. This explains why old memories, once consolidated, may survive even when hippocampal function is compromised. In contrast, recent events, still dependent on hippocampal circuits, may be lost if the hippocampus is damaged. The distinction highlights the layered nature of memory storage, with different brain regions contributing at different timescales. This transfer highlights the complementary strengths of different systems. The hippocampus excels at rapid encoding, capturing new experiences quickly. The cortex provides stability, integrating memories into broader knowledge over longer timescales. Together, they balance flexibility with durability, ensuring that new information can be acquired without erasing the old. Not all memory, however, relies on the hippocampus. Procedural memory, which supports skills and habits, depends on circuits in the basal ganglia and cerebellum. Learning to ride a bicycle or play an instrument recruits these networks, which gradually automate sequences of movement until they can be performed with little conscious thought. Emotional memory engages the amygdala, which tags events with significance. Its influence explains why emotionally charged events are often remembered more vividly than neutral ones. Working memory, by contrast, operates on a much shorter timescale, holding information briefly for immediate use. Prefrontal cortex circuits sustain activity through recurrent connections, allowing one to keep a phone number in mind long enough to dial it or to follow multi-step instructions. Each type of memory reflects specialized networks, yet all interact to create the richness of experience. The fragility of memory is as striking as its power. Memories can fade with time, be altered during retrieval, or even be influenced by suggestion. Each act of recall is also an act of reconstruction, susceptible to change. Rather than a faithful recording, memory is more like a story rewritten each time it is told. Yet this plasticity has adaptive value. It allows memories to be updated with new information, keeping them relevant to current contexts. If a familiar route changes due to construction, memory can be revised to reflect the new path. Similarly, reinterpretation of past events in light of new experiences can shift their emotional tone. Memory is not a fixed archive but a living system, continuously reshaped by use. Emotion strengthens memory through the amygdala's influence on hippocampal circuits. Events that carry emotional weight are more likely to be remembered vividly, and for longer periods. This bias ensures that significant experiences, those tied to survival, social bonds, or personal meaning, leave lasting marks. At the same time, excessive emotional arousal can impair memory, narrowing focus or disrupting consolidation. Under extreme stress, the flood of stress hormones can interfere with hippocampal encoding, leading to fragmented or distorted recollections. This dual role illustrates the importance of balance. Emotion guides memory toward relevance but can overwhelm it when unchecked. Reflecting on memory reveals its role in continuity of self. Without it, life would be a series of disconnected moments, with no story linking past to present. With it, experiences accumulate, shaping identity and guiding future choices. The hippocampus and cortex, through their dialogue, transform daily events into the narratives that define us. Memory supports not only personal identity, but also cultural continuity. Collective memories, passed down through stories and shared experiences, bind groups together. These collective narratives rely on individual brains but transcend them, illustrating the social dimension of memory. Several specialized forms illustrate the diversity of systems. Semantic memory stores facts and general knowledge, independent of the context in which they were learned. It allows us to know that Paris is a capital, or that water boils when heated, even if we cannot recall when we learned these facts. Episodic memory preserves specific events situated in time and place, such as recalling a birthday party or a first day at school. The hippocampus is especially vital for this form. Prospective memory supports remembering to perform future actions, such as taking medicine or keeping an appointment. This type involves prefrontal circuits coordinating intention with future cues. Autobiographical memory blends episodic and semantic forms, creating a personal narrative that defines identity. These distinctions highlight that memory is not monolithic but multifaceted, each type relying on distinct yet overlapping circuits. Memory systems can be disrupted in specific ways. Damage to the hippocampus produces anterograde amnesia, preventing new memories from forming while sparing older consolidated ones. Degeneration of cortical networks in Alzheimer's disease erodes both recent and remote memory, eventually fracturing identity itself. Disorders of working memory, often linked to prefrontal dysfunction, impair the ability to hold and manipulate information, affecting reasoning and planning. Yet even here, plasticity offers resilience. Other circuits may compensate, and strategies such as external reminders can support function. Rehabilitation often harnesses remaining strengths, emphasizing the adaptability of the memory system even in the face of loss. Memory's complexity mirrors the complexity of the brain itself. It is layered, distributed, and dynamic, shaped by both biology and experience. It binds together sensation, emotion, and thought, creating continuity across time. Its fragility reminds us of its biological roots, while its resilience demonstrates the adaptability of neural systems. As this section closes, imagine the hippocampus as a scribe, quickly capturing notes of experience, and the cortex as an archive, preserving them in long shelves of knowledge. Around them, the amygdala adds exclamation points of emotion, the prefrontal cortex organizes themes, and the basal ganglia engrave habits into durable patterns. Together they compose the book of a life never finished, always edited, but rich with meaning. In the next section, we will turn to decision networks, exploring how circuits in the striatum and cortex guide choices, risks, and habits. Decision-making may feel like a purely conscious act, yet it emerges from circuits that weigh options, evaluate risks, and compare outcomes long before awareness. These processes involve an interplay between the prefrontal cortex, striatum, and limbic structures, each contributing a distinct perspective. Together, they create a system that balances immediate rewards with long-term goals, shaping choices from the mundane to the profound. The prefrontal cortex acts as a planner and evaluator. It holds possible scenarios in working memory, simulates consequences, and integrates them with current goals. Different subdivisions specialize in different aspects. The dorsolateral prefrontal cortex helps maintain and manipulate information, supporting logical reasoning. The ventromedial prefrontal cortex assigns value, integrating emotional and motivational signals. These regions interact constantly, creating a flexible framework for adaptive choice. The striatum, part of the basal ganglia, provides another perspective. It learns from past outcomes through reinforcement, strengthening connections for actions that have led to rewards. Dopamine plays a key role here, signaling the difference between expected and actual outcomes. When results are better than predicted, dopamine surges, reinforcing the action. When worse, dopamine dips, weakening the connection. Over time, these adjustments create habits and preferences that guide future behavior. The limbic system contributes emotional and motivational weight. The amygdala flags options associated with threat or reward, biasing attention. The hippocampus provides memory of context, recalling whether similar situations led to success or failure. These influences ensure that decisions are not abstract calculations but grounded in lived experience and feeling. Importantly, decision circuits operate on multiple timescales. Fast, automatic choices arise from well-learned habits encoded in the striatum. Slower, deliberative choices involve prefrontal simulation of outcomes. The brain shifts between these modes depending on context. In familiar situations, habits dominate, conserving effort. In novel or high-stakes settings, deliberation takes precedence, allowing careful evaluation. Risk and uncertainty engage these systems intensely. When outcomes are uncertain, the prefrontal cortex weighs probabilities, while the striatum tracks potential payoffs. Different individuals show different balances, reflected in tendencies toward caution or risk-taking. These tendencies are shaped by both genetics and experience, as repeated exposure to reward or loss tunes the sensitivity of circuits. Decision-making also relies on control systems that inhibit impulses. The prefrontal cortex can suppress actions suggested by habit or emotion if they conflict with long-term goals. This control is not absolute but effortful, requiring attention and energy. Fatigue, stress, or distraction can weaken it, allowing impulsive choices to slip through. The balance between impulse and control is central to human behavior. At the population level of neurons, decision circuits integrate signals from many sources. Some cells encode value, firing more for preferred options. Others encode probability or uncertainty. Still others represent chosen actions. Together, these populations form a dynamic competition, with the eventual choice emerging from the option that gains the strongest support. Reflecting on decision-making reveals its layered complexity. A simple act, such as selecting a meal, may involve memory of past experiences, evaluation of current needs, anticipation of future consequences, and emotional preferences. The outcome feels immediate, yet it is the product of distributed computation across multiple brain regions. Habits illustrate the power of reinforcement learning. Once established, they can guide behavior with little conscious input, freeing attention for other tasks. Yet habits can also become rigid, persisting even when circumstances change. Flexibility depends on the ability of prefrontal circuits to override habitual striatal patterns, a capacity that varies with context and state. In social contexts, decisions incorporate additional layers. Prefrontal and limbic circuits evaluate not only personal outcomes, but also social norms, reputation, and fairness. Brain regions such as the anterior cingulate cortex monitor conflict between self-interest and social obligation, shaping behavior in cooperative settings. This integration highlights the social dimension of human decision-making. The vulnerabilities of these systems become evident when balance is lost. Excessive striatal drive may lead to compulsive habits, while impaired prefrontal control may foster impulsivity. Dysregulated dopamine signaling can skew evaluation of reward, biasing decisions toward risk or reducing motivation. These examples underscore the importance of balance across circuits for adaptive choice. As this section closes, Imagine decision-making as a dialogue between three voices: the striatum, recalling past rewards; the limbic system, coloring options with emotion; and the prefrontal cortex, projecting consequences into the future. When they harmonize, choices are adaptive. When one dominates unchecked, decisions may falter. In the next section, we will explore how these same circuits can become imbalanced, leading to disorders that illuminate the fragile equilibrium of the brain. Disorders of the nervous system reveal what happens when balance is lost in circuits that normally sustain perception, movement, and thought. Studying these conditions is not only a clinical concern, but also a window into function. When a system breaks, its failure highlights what it once contributed. Through this lens, neurological and psychiatric disorders provide profound insight into how the brain works when healthy. Neurodegenerative diseases illustrate the vulnerability of specific neurons and pathways. In Parkinson's disease, dopamine-producing neurons in the substantia nigra gradually degenerate. As dopamine levels fall, the basal ganglia lose their ability to regulate movement smoothly. Tremor, rigidity, and slowed motion emerge as visible signs of deeper dysfunction. This selective loss shows how crucial dopamine is to motor control and motivation. In Alzheimer's disease, degeneration spreads through the hippocampus and cortex, disrupting memory and cognition. Abnormal protein deposits accumulate, synapses weaken, and circuits break down. The progressive loss of episodic memory reflects the hippocampus's role in encoding, while later deficits reveal the importance of cortical integration. Alzheimer's demonstrates how memory is not stored in one place, but distributed across networks. Other conditions highlight the importance of communication between neurons. In multiple sclerosis, immune attacks on myelin impair conduction along axons. Signals slow or fail, leading to weakness, sensory changes, and fatigue. The disorder reveals how essential insulation is for rapid and reliable transmission. Without it, the efficiency of long-distance communication collapses. Epilepsy provides another perspective. Here, excessive synchronization of neuronal firing produces seizures. Circuits that normally balance excitation and inhibition become destabilized, allowing runaway activity. The condition illustrates the importance of inhibitory control in preventing chaos. It also shows how oscillations, normally adaptive, can become pathological when unchecked. Psychiatric disorders reflect imbalances not of structure alone, but of function and regulation. In depression, circuits involving prefrontal cortex, amygdala, and reward pathways show altered activity. Motivation and mood shift, not because neurons are absent, but because patterns of signaling change. Anxiety involves hyperreactivity of the amygdala and insufficient regulation from prefrontal regions, producing persistent vigilance even in safe settings. These conditions reveal how emotion depends on dynamic balance between reactivity and control. Schizophrenia highlights the role of neurotransmitter systems in shaping thought. Altered dopamine and glutamate signaling disrupt circuits linking cortex and subcortex, leading to hallucinations, delusions, and disorganized cognition. The disorder shows how neurotransmitters do more than modulate, they structure the very coherence of perception and belief. Stroke provides yet another example. When blood supply to a region of brain tissue is interrupted, neurons deprived of oxygen and nutrients die. The functions supported by that region, speech, movement, vision, may suddenly vanish, yet plasticity allows partial recovery, as neighboring regions reorganize to compensate. Stroke therefore reveals both the fragility and resilience of neural networks. Studying these disorders underscores recurring themes. Genes, neurotransmitters, synapses, and circuits all contribute to vulnerability. A mutation may alter a protein, an immune attack may strip insulation, an imbalance of excitation and inhibition may destabilize firing. In each case, the consequence is not abstract, but deeply human. Loss of movement, memory, mood, or self. Yet disorders also reveal the adaptability of the brain. Rehabilitation after injury, compensation by spared circuits, and plasticity in response to change all demonstrate resilience. Even in the face of loss, networks attempt to reorganize, finding new pathways to sustain function. This capacity for adaptation is as central to neuroscience as vulnerability. Reflecting on these conditions invites both caution and curiosity. They remind us that the brain's complexity brings both strength and fragility. Small disruptions can cascade into profound effects, but the same plasticity that enables learning also supports recovery. Disorders are not only challenges to health, but lessons about the principles that govern all brains. As this section closes, Imagine the nervous system as a vast network of connections, resilient yet vulnerable. Break one link, and the pattern shifts, weaken many, and the system falters. In the next section, we will turn from pathology to development, tracing how the nervous system takes shape from early beginnings to mature function. Development of the nervous system is a journey from a simple sheet of cells to a vast network of billions of neurons, each connected in intricate patterns. This process unfolds in stages: proliferation, migration, differentiation, synapse formation, and pruning, each governed by molecular signals and activity-dependent refinement. Though the outcome is a mature brain, the path reveals principles that resonate throughout neuroscience: growth constrained by guidance, diversity emerging from simple origins, and refinement through use. The earliest stage begins within the embryo. A flat sheet of ectodermal cells folds inward to form the neural tube, the precursor of the brain and spinal cord. This structure, though primitive, contains within it the blueprint for regional specialization. Molecular gradients along its length instruct cells to adopt identities, producing forebrain, midbrain, hindbrain, and spinal cord. Even at this stage, patterning lays the foundation for future circuits. Proliferation follows, as stem-like progenitor cells divide to produce neurons and glia. Waves of cell division expand the population, filling the developing brain with precursors. Yet proliferation is tightly regulated. Too much or too little can disrupt structure. Each progenitor has intrinsic programs and responds to environmental signals, balancing growth with restraint. This balance ensures both sufficient numbers and controlled architecture. Once generated, neurons embark on migration. Guided by molecular cues and glial scaffolds, they travel from their birthplace near the ventricles to their final destinations. In the cortex, neurons move outward, forming layers in an inside-out pattern, where later-born neurons pass earlier ones to settle nearer the surface. This migration creates the stratified architecture characteristic of the cerebral cortex, with each layer supporting distinct functions. Differentiation, then, gives neurons their unique identities. Some become excitatory pyramidal cells, projecting long axons to distant targets. Others become inhibitory interneurons, shaping local circuits. Gene expression patterns combined with local signals direct these fates. The diversity of neuronal types, from sensory relay cells to motor neurons, reflects the interplay of intrinsic programs and external influences. Axons extend toward their targets, guided by chemical gradients and contact cues. Growth cones at their tips sense the environment, steering toward attractants or away from repellents. This guidance ensures that connections form in the correct regions, linking retina to thalamus, thalamus to cortex, and spinal motor neurons to muscles. Errors occur, but redundant cues and pruning refine the wiring. The precision of these pathways highlights the power of developmental guidance systems. Synapse formation marks the next stage. When axons reach their targets, they form synaptic contacts, releasing neurotransmitters and inducing receptors in postsynaptic cells. At first, synapses are exuberant, with more connections formed than will persist. This overproduction ensures coverage and redundancy, allowing activity to select the most useful links. Synaptic pruning follows, eliminating weaker or less active connections while strengthening others. This sculpting refines circuits, creating efficient networks adapted to experience. Activity plays a crucial role in refinement. Neurons that fire together strengthen their connections, while those that do not lose influence. This principle, seen in Hebbian plasticity, operates during development to shape circuits according to use. In the visual system, for example, input from both eyes competes for territory in the cortex. Balanced activity produces coordinated maps, while imbalance, such as from strabismus, can skew representation. Experience therefore directs structure, embedding the environment into the architecture of the brain. Glial cells also contribute to development. Radial glia provide scaffolding for migration, astrocytes regulate synapse formation, and microglia prune connections. Their roles reveal that glia are not passive supports but active participants in wiring. By shaping which synapses persist and which are removed, glia influence the very structure of networks. Critical periods highlight the timing of plasticity. During these windows, circuits are especially sensitive to experience. Language acquisition, visual alignment, and social bonding all depend on timely input. If experience is absent or distorted during a critical period, development may be altered permanently. Yet outside these windows, plasticity persists, though with different rules and reduced flexibility. Critical periods demonstrate the interplay of intrinsic timing and environmental shaping. Development continues into adolescence and adulthood. Synaptic pruning refines prefrontal circuits into the second decade of life, supporting maturation of judgment and impulse control. Myelination also proceeds, increasing conduction speed and efficiency. These changes reflect not only growth but also specialization, as circuits adapt to the demands of adult life. The vulnerabilities of development are profound. Disruptions at any stage, genetic mutations, environmental toxins, or deprivation of experience can alter outcomes. Some disorders reflect excessive proliferation, others failed migration, others disrupted pruning, yet resilience also exists. Plasticity and redundancy allow compensation, enabling function even when early pathways are compromised. Development is thus both fragile and robust, a balance that echoes throughout neuroscience. Reflecting on development reveals themes of emergence and refinement. From a simple tube grows an organ of staggering complexity. From overproduction arises efficient networks. From experience comes adaptation, embedding environment into biology. Development is not only a story of growth but of sculpting, where potential becomes structure through interaction with the world. To expand further, consider how development recapitulates principles of evolution. The layered structure of the brain reflects ancestral stages: brainstem circuits for survival, midbrain for orientation, cortex for complex integration. In the embryo, these stages appear sequentially, as if replaying evolutionary history. Development thus bridges the scale from individual growth to species lineage. Another expansion lies in the lifelong echoes of early experience. Stress, enrichment, or deprivation in childhood can alter circuits that persist into adulthood. Sensitive periods leave marks that shape personality, cognition, and resilience. These enduring effects show that development is not merely preparation for adulthood, but a continuous influence on the trajectory of life. Finally, development illustrates the inseparability of biology and environment. Genes provide instructions, but activity and experience shape outcomes. A child raised with language develops circuits for speech. One deprived may struggle even with later exposure. The brain is built to be molded, expecting input to complete its formation. This openness underscores the interactive nature of neural development. As this extended section closes, imagine the nervous system as a vast sculpture, beginning as raw stone. Proliferation provides material, migration positions it, differentiation shapes it, synaptogenesis connects it, pruning refines it, and experience etches final details. The sculpture is never fully complete, always subject to small adjustments, yet its form emerges from this interplay of growth and guidance. In the next and final main section, we will examine the nervous system at its largest scale, considering how networks spanning the whole brain coordinate into the phenomena we call consciousness. Consciousness is often described as the most elusive subject in neuroscience. Unlike vision or movement, which can be traced through pathways and measured in clear outputs, consciousness refers to the subjective quality of experience, the sense of being aware, of having thoughts, feelings, and perceptions unified into a continuous stream. The challenge lies in linking neural activity, which can be observed, to this inner world which cannot be directly measured but only reported. One starting point is the concept of global integration. Neural circuits operate locally, processing specific tasks in specialized regions. Yet conscious awareness seems to require that these distributed activities be bound together into a coherent whole. Theories such as global workspace models propose that consciousness arises when information in one region is broadcast across widespread networks, allowing perception or thought to become globally available. In this view, consciousness is not everywhere in the brain, but emerges when signals cross thresholds of integration. Oscillations play a central role in this integration. Synchrony across distant regions allows information to be shared efficiently, binding features into unified experience. For example, when you perceive a face, neurons in the visual cortex process shape, color, and motion separately, yet conscious recognition requires that these be combined. Oscillatory coherence may provide the timing that links them, ensuring that they are experienced as one object rather than fragments. Attention also interacts with consciousness. Many stimuli are processed unconsciously, influencing behavior without awareness. Attention seems to act as a gate, allowing selected information to enter conscious report. Yet attention is not identical to consciousness. One can be aware of a stimulus without focusing on it, and one can attend to something without full awareness. The relationship is intimate, but not reducible to one-to-one equivalence. Theories diverge in explaining the mechanisms. Integrated information theory suggests that consciousness corresponds to the capacity of a system to integrate information in a way that is irreducible. According to this view, the richness of experience reflects the high degree of integration in cortical networks. Other perspectives emphasize recurrent processing, where feedback loops between cortical layers sustain activity long enough to support awareness. Each theory captures aspects of the phenomenon, but none provides a full account. Neuroanatomy provides clues about where consciousness is supported. The cortex, especially frontoparietal networks, appears essential for reportable awareness. Lesions in these regions can abolish or alter consciousness, producing neglect or unawareness of stimuli. The thalamus also plays a crucial role, serving as a hub that coordinates cortical communication. Damage here can lead to profound disruptions of consciousness, as in coma or vegetative states. Brainstem structures maintain arousal, without which awareness cannot be sustained. Together, these regions form a hierarchy: brainstem for wakefulness, thalamus for coordination, cortex for content. Disorders of consciousness illustrate these dependencies. In coma, both arousal and awareness are absent, often due to damage in brainstem or thalamic regions. In vegetative states, arousal returns. Patients open their eyes and show sleep-wake cycles, yet awareness remains minimal or absent. In minimally conscious states, fragments of awareness emerge, revealing how fragile and graded the phenomenon can be. These conditions underscore that consciousness is not all or none, but exists along a continuum. Experiments with anesthesia provide further insight. Agents that suppress consciousness often reduce long-range communication between brain regions, even while local activity persists. The cortex may still process inputs, but without global integration, awareness fades. Recovery of consciousness involves restoration of these connections, supporting the idea that widespread communication is essential. Dreaming offers another window. During rapid eye movement sleep, cortical activity is high, resembling waking states, yet the external world is largely disconnected. The vivid experiences of dreams suggest that consciousness does not require sensory input but can arise from internally generated activity. However, the altered logic and instability of dreams reveal the importance of prefrontal control in shaping coherent waking consciousness. Reflecting on consciousness raises philosophical questions. Is it merely an emergent property of neural complexity, or does it involve principles beyond current science? Neuroscience approaches the question by seeking correlations, identifying the neural signatures that accompany awareness. Yet correlation does not fully explain causation. Why should certain patterns of activity feel like experience? This remains the so-called hard problem, linking mechanism to subjectivity. Despite these challenges, practical advances emerge. Identifying neural markers of consciousness aids in assessing patients with impaired awareness, guiding prognosis and care. Understanding the effects of anesthesia improves safety and control. Exploring the neural basis of altered states, from meditation to psychedelics, reveals how flexible consciousness can be, shaped by changes in connectivity and chemistry. Consciousness also interacts with memory, emotion, and identity. Awareness allows reflection, giving memories narrative form and emotions meaning. Without consciousness, experience would be fragmented and inaccessible. With it, we weave a sense of self that persists through time. Circuits that bind perception, attention, and memory are therefore not only functional but foundational to identity. The study of consciousness reveals themes seen throughout neuroscience: integration, balance, and plasticity. Local circuits provide detail, but global networks provide unity. Stability ensures continuity, while flexibility allows change. Consciousness may be the most complex expression of these principles, the emergent property of a system designed to process, integrate, and adapt. Reflecting personally, one may see consciousness as the backdrop against which all else unfolds. It is both the stage and the play, both the container and the content. Neuroscience provides models and mechanisms, but the lived quality remains immediate and irreducible. To study it is to bridge the gap between objective description and subjective experience. As this extended section closes, imagine the brain as a vast city at night. Local neighborhoods buzz with their own activity, but when the lights synchronize across districts, the city becomes visible as a whole. Consciousness is like this illumination, not the activity itself, but the coordination that makes the activity into an experience. In the final wind down, we will let this exploration settle, slowing the rhythm and inviting rest, carrying with us both knowledge and calm.